Okay, so technically we're recording at this point, but I'm going to be able to edit this beginning section. And so Jace, if you are ready, I'm going to say go ahead and just begin your presentation when you are ready and tell me next when you want me to advance.
Okay.
Okay, so whenever you're ready, you can begin anytime starting now.
Ready. Hello, everyone. I want to thank you all for attending this webinar. Next slide, please. I'm Jess Xu, and from European Discovery. I'm technical manager and based in Taiwan. Today, I want to share some of our new approach for carcinogenicity risk assessment, including some AI-assisted and especially for ethical alternatives to traditional animal study. Next slide, please. First, this presentation is based on the FDA guidance on carcinogenicity testing of pharmaceuticals as shown in the slide. In November 2022, FDA issued the guidance S1B revision attended, which introduced a weight of evidence approach to assess carcinogenicity risk in drug discovery. This approach is part of FDA's efforts to reduce animal testing while maintain safety requirements. The slide also highlights 3 key ICH guidelines for carcinogenicity. S1A covers the need for long-term rodent study by defining conditions based on treatment duration and other clinical aspects of the drug. S1B is our major topic today, addressed some testing approach for evaluating the carcinogenicity potential of pharmaceutical by approving integrative weight of evidence criteria to determine whether 2-year rat study add value to human risk assessment, thereby reducing animal testing while redirecting results toward mechanical-based evaluation. And S1B outlines criteria for selecting the dose in carcinogenicity animal study, provide great flexibility in optimized study design. This comprehensive framework guidance pharmaceutical developers in evaluating carcinogenicity risk for new drug candidates. Today, I will discuss about the weight of evidence approaches in our in vitro technical and to accumulate more information in early drug discovery stage. Next, please. For any drug expect to use more than 6 months, carcinogenicity study should be conducted. Traditionally, all carcinogenicity studies follow the guidance established by ICH, which have involved animal studies to determine whether the substance cause tumors according to ICH and OECD guidelines. This study involved hundreds of animals and normally 2-year study and maybe 1 more years for analysis and the data report. Animal number typically consisted of 50 to 65 animals per set per dose level with 3 dose level and double control groups. And at least cost exceeding $2 million. This was considered the most time and resource consuming study in toxicology. However, there's still some limitation for animal studies, such as no suitable prostate cancer animal models, different cellular origins of ovarian tumor between human and rodents, and no parallel pathology lesion for breast cancer in human and mice. However, the authority regulatory also aware this part. In August 2022, the new guidelines S1B introduced a more refined weight of evidence approach. The new frame evaluates several key categories, including drug target, pharmacology, and toxicology study, and the spatial investigation. Later we will discuss the detail. Based on this comprehensive assessment, we can now determine whether a traditional 2-year rate study would add value to understand human carcinogenicity risk. This leads to 3 possible outcomes. You can see in the left lower side of the slide. Unlikely, likely, or uncertain. For whether 2 years rate study would add value, if the risk is unlikely, this suggests the animal study no added value. If the risk is likely, further investigation to rate study also does not add value. Only when the conclusion is uncertain for the risk, insufficient evidence to determine either ways, then a 2-year rate study would add value. Typically, the decision to waive this study depends entirely on the judgment of regulatory authorities in each country, like US FDA, EMA, and other countries' regulatories. Others will make decisions based on the specific features of the drug, risk assessment results, and the available scientific evidence. Next slide, please. This slide presented the critical weight of evidence factors outlined in the guidance for the assessment risk of pharmaceuticals. I directly adapt this paragraph in the left side to let's check one by one. The weight of evidence approach represents a comprehensive assessment methodology that evaluates all relevant carcinogenicity potential data. There are 6 key factors. 1, drug target biology and primary pharmacology, indicating target distribution, including target distribution and the metabolic activity across species. 2, second pharmacology screen that inform of target efforts and binding to receptors, particular carcinogenicity-related targets. 3, histopathology from animal study, particular from 6-month rate study. 4, hormone effect, including endocrine response mechanism. 5, the genotoxicity study. It's also based on ICH guideline S2, including in vitro study like M-test and the micronuclear assays. 6, immunomodulation, which is according to ICH S8 guidance. It is a broader immunosuppression may indicate human risk. Next slide, please. Next. This is also the outline of today's presentation. I will follow these factors one by one to present our new approach for weight of evidence approach for carcinogenicity risk. The right slide of the slide showed how Europeans have developed specialized testing panels aligned with these factors, including safety panels and the carcinogenicity risk assess panel for secondary pharmacology. AI-assisted cell transformation as phenotypic assay for histopathology in vitro assay, and the endocrine system focus panel for hormone effect. In vitro genotoxicity testing are well-known study for IND filing, and in the bottom biomap for immunomodulation assessment. This integrated approach helps us to for the carcinogenicity risk assessment while supporting the 3R principle to reduce animal testing. Next slide, please. Okay, the first factor specifically is drug target biology and the primary pharmacology mechanism of both the parent compound and the major human metabolites. This evaluate including detailed assessment of target distribution across species, pharmacology activity, and the potency, data from genetic engineering, and everything about the primary target. Factor 1, we don't discuss too much, since this is the primary target that requires detailed investigation of this mechanism and carcinogenicity risk by the developer. Next. Secondary pharmacology is crucial weight of evidence factors. It involves screening both the parent compounds and the major metabolites to understand their selectivity and off-target effect. This off-target effect assessment operated on two key principles. First, establish the connection between molecular targets and the carcinogenicity-related pathway. Second, employed in vitro testing against multiple unintended targets to reduce toxicology risk. The main tool for evaluation is the cancer-related off-target panel, which help us identify potential carcinogenicity risk. Next, please. Jacobs published a review article that outlines an integrated strategy for non-genotoxic carcinogens. It highlights that exposure to either native or metabolite of chemicals can trigger various cell response related to carcinogenesis, like inflammation, immune reaction, cell division, and cell injury. If the exposure continues, it can lead to further cell proliferation, transformation, and eventually the development of tumor cells. Because of this, identify the right molecular target could surface new useful models for assess the risk of chemical compounds. This target including range from receptors and enzymes. Here we introduce you carcinogenicity risk assessment panel focused on key targets involved in tumor formation, such as receptor binding assay and enzymatic assay, including some kinase, protease, CYP, and more. Our target selection is based on systemical surveys in the PubMed literature reference, emphasizing the most relevant receptor addressing carcinogenesis risk. Next. Here, I want to share with you the results of our case study in the panel. In this panel, we have conducted screening profiling across 30 different targets for various compounds, including well-known carcinogen, mutagen, and non-carcinogen. Our findings are really promising. As shown in the heat map, different blue shows the strength of binding inhibition, with dark color indicating strong interaction. What's especially notable is that our panel identified all potential carcinogens, but did not detect any mutagen, and the mutagen walked through directly to genotoxic mechanism, like interaction with DNA, not interaction with receptor or enzyme. The top of our chart, like PMA, cyclosporin A, and the triglyceride, shows clear binding pattern across multiple targets. This multiple target profiling help us spot compounds with potential carcinogenetic risk. But compounds like cyclophosphamide, mitomycin, and aflatoxin, which directly interaction with DNA and cause genotoxicity, couldn't be fully identified by our panel. This really shows why it is important to combine different assessment methods, like to combine genotoxicity studies. However, the non-carcinogen at the bottom of our chart, like aspartame and methylparaben, shows very little interaction with our panels, which match their relativity low risk for carcinogenicity. Okay, next slide. Thank you. Notably, our panel was cited in a 2022 Frontier in Toxicology publication by Benson et al., which focused on standardized weight of evidence criteria. In the section discussion cancer-related off-target screening panels, highlighting Eurofins discoveries latest contribution to carcinogenicity screening by providing a structured approach to evaluating cancer risk in drug development. Although a comprehensive list of risk targets is still being developed, Eurofins panel has gained significantly attention, with many international clients, including those advised by the FDA, and placing orders for this panel for carcinogenicity risk screening. We are making progress in the right direction, though there's still some room for improvement. Next slide, please. The third factor focuses on histopathology data. This including finding from a repeating dose toxicity study with particular emphasis on the 6-month rat study. And however, this is an animal study. Here, I want to highlight the cell transformation assay, or you can call it CTA. The BHAS42 CTA is specially designed to predict carcinogenesis of compounds with cell model and phenotypic assays. This is significant because it published in OECD guidelines that can distinguish between tumor promoter and the tumor initiator as an animal alternative method. We are using the mouse BHAS42 fibroblast cell line, which transfect to have high carcinogenic potential for foci formation. This assay allows us to evaluate how chemical affects cell growth and the transformation process. A key advance is that it can detect both genotoxic and non-genotoxic chemicals. These assays are available on TSAR, the tracking system for alternative methods toward regulatory acceptance. We combine this with other component methods as part of our approach. Next slide, please. Looking at the endpoint measure of 2 key parameters of cell transformation assay. First, cell number quantified by staining directly measure cytotoxicity, giving us immediate insight into compound toxicity. Second, we also assess morphology transformation by counting transformed foci, which indicates actual carcinogenic potential through visible cellular change. For endpoint values of transformed using different platforms, in 60 well plates, it counts the actual number of transformed foci in the well, while in 96 wells format, it measures the proportion of well containing transformed foci. And this treatment, the treatment regimen are designed to target different mechanisms of carcinogenesis. For the initiation assays, we plot fewer cell numbers, then apply chemical treatment during the growth phase. This especially detect compound that may initiate carcinogenesis through DNA damage or mutation. For the promotion assays, we begin with higher cell density. After chemical treatment occurring during the stationary phase between day 7 to day 14. This approach targeting the compound that promote the growth of those already transformed cells, not for the causing initial transformation. The graph on the right side displays the cell growth curve, showing the relationship between cell proliferation stage and the treatment windows. Now, note the growth phase from day 0 to 4, followed by the plateau phase form from log day 6 onward. The black and white bars below indicate when treatments are applied in each assay variant. Next slide. Next slide, please. This slide shows the example of conventional method for the cell transformation assay. As you can see, the assay measures both initiation and promotion phase carcinogenesis using non-positive control compounds. On the left, we have MCA for the initiation phase, and on the right, PMA for the promotion assay, both tested in a 6-well plate format. We demonstrated those dose response effect of foci formation using mmG in the initiation test and the 2AAF for the promotion test. The conventional methodology relies heavily on several manual processes: gene sustaining for visualization, foci counting technology, and critically, scientist judgment for interpretation. Key methodological features are indicated, like basophilic staining. You can see on the right below the slide, spindle-shaped cells, and the multi-layers, and the rodent orientation, and the invention properties. These parameters allow us to systematically evaluate cell transformation. Next slide, please. Here, I want to highlight our new approach improve the cell transformation assay using AI-based image analysis. Compare the conventional and advanced methodology side by side. The conventional method presents several operational challenges. It requires 2 expert analysts working with 6 criteria per plate and the compound. Despite the certification as a guidance from the document, this process remains labor intensity, time consuming, and perhaps most critically, subjective in its assessment. These limitations can impact both throughput and reproductive. Our advanced method address these challenges through implementation of AI-based in-kata image analysis software paired with our image system shown in the photograph. The system has been extensively trained on more than 4,000 transformed foci, creating a robust foundation for consistent objective analysis. This method dramatically efficiently improvement is quantified in the table at the bottom of the slide. For a standard analysis of 36 plate, the manual method typically requires between 70 to 80 hours of its expert time. In contrast, our AI-based in-kata system complete the same analysis in just 8 to 9 hours, a nearly 90% reduction in process time. This technological advancement represents a significant leap forward in our histopathology assessment capabilities. By eliminating subjective variability while increasing throughput, we can deliver more consistent, reliable evaluation. Next slide, please. Look at this image. You can see our in-kata analysis module optimization in action. The left column shows fluorescence staining with Horchus in blue transformed foci against normal cell stained with calcium AM in green. The middle and the right column demonstrated how our analyzed module detect this transforming area. We have significantly enhanced our detection capability through our optimized module, which clearly identify transformed cell area, show a screen masked regions with much greater accuracy than our original images. We use the Pearson correlation coefficient analysis tool, evaluating the linear relationship between two sets of data. In the right side, you can see the figure. When the correlation effect, the factor more than 0.8 indicates a stronger positive linear correlations. And regarding to our assays, the Pearson correlation of our foci counting between scientists and the Incata analyzed module is 0.9517, indicating a strong positive correlation. This suggested that Incata analyzed module can serve a reliable alternative method for foci counting. Next slide, please. Here are the histopathology results. Compare case study finding with literatures. This slide showcases our examination of chemical carcinogens used the AI-assisted cell transformation assay. Among known carcinogens, benzopyrene demonstrated clear initiation active in both our case study and reference. And albitic acid shows promotion activity that align with reference data. However, cyclosporine A and the ranitidine shows promotion activity in our study, but literature finding something different. And then among the, again, MNG and acetylaminofluorine demonstrated consistent alignment with established reference data across both experimental models. In the red row, you can see the positive control chemical perform as expected. MCA confirmed initiation capabilities, while PMA validated our promotion models. Both control align with lecture findings, reinforcing our approach. And in the green row, caffeine and mannitol is the negative control, consistently show no carcinogenic genetic activity across all evaluation. These results validate the AI-assisted cell transformation as a reliable method for objectively assessing carcinogenic potential through both initiation and promotion stages. We will keep improving this assay protocol to make it more robust and scientifically convincing. Next slide, please. Moving to the number 4 factors, we will explore the hormone effect framework. Looking at the slide, the key data source to form a systemically approach evaluates a hormone effect. You can see on the slide. First, the repeating dose toxicity study examining both gross and microscopic changes in endocrine and reproductive organs. This finding provides signals of potential hormone disruption. Second, reproductive toxicology studies, when available, which offers reveal additional histopathological changes related to endocrine function. Third, the compound primary pharmacological mechanism. Understanding whether a drug directly interacts with a hormone pathway or receptors is crucial for predicting potential carcinogenic risk through the endocrine disruption. And the fourth, we assess secondary pharmacology data. for off-target activities that might affect the endocrine system. This unexpected interaction can sometimes be more concerning than the primary mechanism. As we show in the central part of the slide, this data streams coverage to inform the hormone assessment. Next slide, please. Here, we focus on the in vitro way for the nuclear hormone receptors, or NHR, which play critical roles in endocrine-related carcinogenesis. As highlighted in the review 2000, multiple cancer types involve hormone disruption mechanism, including breast cancer, endometrial cancer, ovarian cancer, prostate cancer, and other cancers. On the right, you can see another review also showed physiological expression of nuclear hormone receptor throughout the body. Note that color coding green indicates tumor inhibiting receptor, and red shows tumor promoting receptors, and the blue design those with dual natures. This distribution helps us understanding why certain tissues are particularly affected by the endocrine-mediated carcinogenesis. At Eurofins, we have developed specialized screening panels to evaluate interaction with these critical targets. Our endocrine pharmacological panel, PP321 and the P402 panels. This panel shows us to systemically screen compounds for potential interaction with endocrine targets in the early stage of drug discovery. Next slide, please. And here you can see our case study. This is a detailed heat map illustrating how these compounds interact with various nuclear hormone receptors. The blue bars indicate inhibition, while orange shows activation. Let's highlight some particularly significant interaction. First, CITCO and obeticholic acid demonstrated distinguished pattern of nuclear receptor activation, with particularly strong effect on CAR and FXR, respectively. These two compounds specifically binding to their primary target without promiscuity to other NHRs. You can see on the case study. The profile, like acetylmethylfluorine and mitomycin C, reveal that some mutagens does not interact with their nuclear receptor. Again, they interact with DNA, not receptor. In contrast, low triglyceridone is PPAR gamma receptor activator. It modulates not only PPARs, but also steroid reductase and androgen receptors. And for diacylstilbestrol also shows extensive demodulation of estrogen receptor as well as androgen receptor and the CAR receptor, which explain its potential estrogenic effects and its carcinogenicity potential. Bisphenol A, a compound of significant environmental concern, which is a well-known environment hormone, shows a diverse binding profile with estrogen and androgen receptor activity, supporting concern about its endocrine disruption potential. And some cosmetic preservatives like methyl isothiazolinone does not modulate this receptor. Again, this shows its low carcinogenesis risk. These binding profiles provide crucial insight into how these compounds may influence hormone signaling pathway. By understanding these molecular mechanisms, we can better predict potential toxicology outcomes, carcinogenicity risk, and evaluating environmental impact. Next slide, please. Okay. Number 5, the genotoxicity testing is another critical factors in weight of evidence approach. This test, the testing method, including both in vitro and in vivo methods, all follows OECD guidelines and listed on the table for your reference. For in vitro, we utilize the M bacterial reverse mutation assay follow OECD 471, and which detect DNA point mutation. And the in vitro micronucleus testing under OECD 487 identifies chromosome damage leading to micronuclear formation. And the slide, you can see the related assay number for your reference. And as these in vitro genotoxicity assays are conventional requirement for IND submission and well known with those pharmaceutical industrials, we will provide only a brief overview. Next slide, please. Here we see the last point 6 on our slide. Evidence of immunomodulation is evaluated in the accordance with ICH S1B. And this is particularly important because evidence of a broader immunosuppression may provide sufficient concern for human carcinogenicity risk. At European Discovery, we utilize the biomap diversity plus human primary cell-based translational profiling platform to assess potential immunomodulation effects. The advanced platform features 12 systems with 148 total endpoints, allow us to capture a broader range of human biology response. The platform organized into various key biological domains such as vascular, fibrosis, immune, and the respiratory system. What makes this approach particularly valuable is that the stimulation condition effective model adaptive response like inflammation, immune response, tissue remodeling, and the wound healing process. And we will use this method for the immunomodulation potential and associated risk. Next slide, please. Biomap creating a phenotypic fingerprint of modulation. Here we can see how it works. We use human primary cell from at least 3 healthy donors, cultured at lower passages, and stimulate with disease-related cytokines. This allows us to model complex disease biology and measure the precise impacts of tested agents on the protein-based biomarker. These 148 protein-based readouts captured a comprehensive view across multiple biological contexts. By analyzing these readouts, we can detect critical challenges, monitor everything about the cell morphology. It also provides quantitative insight into how tested agents interact with immune system, offering a review of potential carcinogenicity risk. Next slide, please. Next. Biomap compares data from drugs with similar adverse reactions using a comprehensive database of over 4,500 drugs. We map out 9 critical toxicity domains: acute toxicity, immunosuppression, skin irritation, liver toxicity, organ toxicity, skin rash, thrombosis, and vascular toxicity. It is a 3-step approach. First, we compare drug profiling to identify shared biological activities. Then we pinpoint common molecular interaction. Finally, we confirm our finding through data mining, supporting our hypothesis with both statistically and biological evidence. Next slide. Here is a case study using cyclosporin A. In the upper portion is comprehensive phenotypic fingerprint across multiple cellular systems. Each column represented different cellular models. The colored lines represents cyclosporin A's effect at different concentration on various biomarkers. And the downward inhibitions, specifically in the SAG system and the BT system. This pattern reveals cyclosporin A is a very strong immunosuppressive. The lower portion of the slide shows immunosuppression clearly identified at the concentration as low as 40 nanomolar. This immunosuppression effect directly connects to carcinogenesis through the impairment of immunomechanism. When these pathways are inhibited, the body's ability to recognize and eliminate neoplastic cells is reduced. So the tumor cell might escape from our immune system. This approach provides critical earlier insight into potential immunomodulation effects that might contribute to the carcinogenicity risk. Next slide, please. Okay, we have some summaries of our approaches. This slide illustrates how we integrate key weight of evidence factors to assess human carcinogenetic risk according to the guidelines. The framework helps determine a 2-year rat study is more or less likely to be necessary. There are 6 critical factors, as I just mentioned, are evaluated. Target biology, secondary pharmacology, histopathology, hormone effect, and the genotoxicity, and the last one, immuno effect. For each factor, the assessment moves along a spectrum. From the left side indicates conditions where traditional 2-year studies are more likely needed because the chemical with a poorly characterized pathway, higher of target effect, or higher concern other findings. And to the right side shows the less likely for the 2-year animal study, as the chemical with well-known biology, highly selectivity, and no concerning findings. This integrated approach allows for more informed decision-making about resource-intensive carcinogenicity testing, I mean animal study. Next slide, please. Okay, this is our final slide, something like takeaway home message. Our final slide demonstrates how this weight of evidence factors are implemented across the entire drug discovery pipelines. The timeline shows how assessment evolved. Our key innovation approach, including in the lead identification and optimization process or stage. Like target-based carcinogenicity panels and the AI-based cell transformation assay is for histology, pathology. Nuclear hormone receptor endocrine panels specifically for hormone effect. Biomap for immunomodulation screening. The last thing I want to emphasize here is the integration of these factors shows an advancement in carcinogenicity risk assessment by combining mechanism understanding of receptor interaction with other key determinants and follow up by phenotypic studies like some AI-powered biomap or cell transformation assay. We can make more informed decision about compound safety while supporting the 3R principle of animal testing reduction. Next slide. Thank you for your attention. I'm happy to address any question about this integrated approach. Thank you.
Okay, perfect.
Okay, thank you.
Perfect. How did you feel about that?
A little bit nervous, so some pronunciation is not correct.
Well, no, you were awesome. Me, however, I advanced a couple of your slides incorrectly, so I'm going to try to edit those in my
Okay,
edits.
thank you.
But I think you did amazing, Alexander and Joy. I don't know if you wanted to come on, if you had any
Yeah, you did a
comments.
great job, Jay. I think the storytelling is really compelling. And you know, I'm not as technical as some of the audience members will be, so I know they're going to get even more out of it.
Okay, okay, great. Thank you, thank you. Okay, and I have a question for Alice. And you mail me to. I have to draft some question.
Yes, some
For
seed questions. So we will send that to Wendy and the drug target review. Typically, how it works is we have 3 to 5 questions that we've already created, and then Wendy will just read them off as if they came from the chat. So the people on the call will have no idea that no one asked them that we did. And if that's basically like, if there's a question where you want to go into more detail on a certain thing, that's kind of the point of the seed questions.
So I answer the question during the Q&A section myself.
Correct.
Correct. Yep.
Okay. Okay. Thank you. I got it. I'll try to mail to you by the end of this week. Thank you.
Perfect.
Awesome. And then I'll work on the very few edits that I need to do for the recording, and then I'll provide those back to Drug Target Review. And then I believe, Alex, they'll include you and Joy on that final recording just as a final approval.
Perfect.
And of course, and then once we have that final approval, I'll load that, or they'll load that to the final platform, and we'll test that recording ahead of time just to make sure the day of the live event that we're good. And so the day of the live event, we do still ask that each of you and Jace, if you would join us 30 minutes prior to going live. So that would be it, depending on where you are. For me, I'm in the Eastern time zone. Jace, where is Jace? Where are you?
In Taiwan. I'm
You're
not
in
sure.
Taiwan.
Yeah, I'm not sure my time zone.
I'm not sure of your time zone either, but if you would join us 30 minutes prior to us going live, that way we can just do your sound check, make sure everything is good. And
you
again,
you mean on the real webinar day before
correct.
the webinar? Okay, I can join I can join the meeting. Yeah.
Okay, perfect. Yep, and that's April 2nd, I believe.
Okay. Okay. For me, it's 9 p.m. Yeah. Okay.
9 p.m. Okay.
Okay.
So yes, if you would join in that 30 minutes before, that would be perfect. And so that would probably be, so you would join us at 8:30, I guess?
I'm joining 9 p.m. and the 9:30 will start the webinar.
Got it. Okay.
Okay. Okay.
All right. Awesome. Awesome. So any other questions, Alex, Joy, Jace, anybody?
Nothing from my end. I guess the next steps are just, looking at the recording, making sure everything looks good and sounds good, and just getting those seed questions to you. So unless Joy has anything else, I think we're in good shape.
Yeah,
awesome.
Sounds good.
Awesome.
And I can download the record also.
I believe we'll provide the recording to each of you just for a quick review.
Okay, thank you. Got it.
Perfect. Perfect. Okay, so if there's nothing else, then I should have that recording available by about Friday. That still gives us plenty of time. And if you guys have any questions, Alex, I believe you enjoy both have my email as well. Feel free to email me or John at DTR. And I think we are good.
Perfect. Thank you, Wendy.
Great. Thank you,
Thank you, Jason,
everyone.
for your work today.
Thank you. Alex, thank you.
Perfect. Thank you so much. Have a good evening or afternoon, depending on where you are.
Okay, thank you.
Take care.
Bye.
Great.
Take care.
Bye,
Bye-bye.
everyone.
Bye, everyone.